Hi there, and I would like to do is go ahead and start our review for the AP exam. What I have for you is a 3 units on the membranes, the macromolecules, and the organelles and the energetic organelles. Okay, so let's begin. Okay, so what we want to do is look at polymers and macromolecules. And as you can see at the question at the top, we really need to understand a little bit about the components that make up these macromolecules. Now, the three that are listed, the first three, amino acids, nucleotides, and macromolecules are all true polymers. The fat molecules are not truly polymers, but they are macromolecules, and they're very important to living things. So a quick review, you know that the amino acids connected together make a polypeptide chain. Those polypeptide chains can then make proteins into quaternary structures. The nucleotides combine together to make DNA or RNA. DNA makes a double helix, RNA makes a single side. The monosaccharides are sugars, and then they can connect in a variety of ways, and they make your starches and polysaccharides. Now, the fats are made up of glycerides or glyceraldehydes, which are 3 carbon sugars, or glycerol, and the fatty acids. And they can be made up of what are called triglycerides, which are the 3 fatty acids connected together. So what we want to do is begin this discussion of how we can then look at those subcomponents and predict what would happen if you changed one of those macromolecules or those single monomers. And the second part is then to talk about that order or sequence and how that affects the properties of the polymers. Okay, so here's an example of 4 polysaccharides. Cellulose, starch, and glycogen are all the polysaccharides, and chitin, which is found in the exoskeletons of arthropods, have nitrogen source connected to the sugars, and you can see they're a little bit different arrangement and type, and that chitin becomes a very hard material that makes up that exoskeleton. So if you look at cellulose and you look at cellulose and you're going to look at starch, you're going to see it has a different kind of bond. And this is the difference. Bonds allow for cellulose to become a structural polysaccharide that allows for the sugar to make up the cell walls, whereas starches and glycogens are really used for the processing and storing of monosaccharides for energy. So here we have a classic example of how you can change the type of bond between them, and it changes their structure. Starch makes a more tubular shape, and glycogen makes a more branched shape. All right, here's the polynucleotides. Quick overview. Here's DNA on one side, RNA on the other. They're anti-parallel. DNA has 4 kinds of nucleotides: adenine, guanine, cytosine, and thymine. And the RNA has adenine, cytosine, guanine, and uracil. And the thymine and the uracil are the 2 different pyrimidines, and the purines, or you remember, are the double rings, and that's guanine and adenine. The deoxyribose is missing that sugar here at the 2' end, and here it is on the ribose side, and the phosphoric acid makes up the sides. And now let's focus a little bit on polypeptides. Okay, so you got your primary structure, which is a polypeptide sequence, which is connected together between the amine group and the carboxyl group. Then you get your secondary protein structure with the 2 examples we've talked about, the pleated sheets and alpha helix. You combine those 2 together, as well as some environmental conditions that allows for the folding and bending. You have your hydrophilic ends on the outside, and then you're going to have your hydrophobics on the inside. If you then take the tertiary structures and combine them together, you get the globular or quaternary structures. Okay, so let's now take a look at membranes. We're going to focus a little bit on this signal transduction. So this signal transduction has these multiple steps, You remember there's a ligand or something on the outside of the membrane. Then there's some kind of process through the membrane, and then some kind of response internally. I want to highlight a whole bunch of different things here as a quick review, but remember we have a signal that then causes the cells to respond, usually in the making of proteins or the controlling of proteins. And because these are multi-step pathways, a very great opportunity, as it says, to regulate and control. So here's that overview. You have the reception, which is the growth factor in this example, bind to receptor on the cell membrane. There's a signal transduction. In this case, this is a cyclic AMP phosphorylation cascade, which then have the ability to take and make what are called kinases that are going to then impact transcription or translation. In this example, it is activating the transcription factor. So things I'd like you to know is to understand that you can change the signal transduction. It's going to have some different response. You need to understand a little bit about how that signal transduction alters that cellular response and describe it, and construct an explanation how certain drugs affect that. So let's go ahead and do the last one. And here it is. This is the overview. You have your epinephrine binds to the protein on the membrane. That's the signal part. The transduction is the activation of the G protein in the cyclic AMP, which then causes the protein kinase to be activated and has a cellular response. In this example, what you see here is the chemical found in marijuana called THC. As it binds to the membrane protein, it goes through the G protein through these kinase products and then affects transcription. In this case, it transcribes this gene called COX-2. COX-2 then is made and then allows for this protein channel to form, which then binds and blocks this synapse where you have glutamine being released between the end of one neuron and then to this dendrite of the next one. And because of this blocking of these channels, it has the effect of slowing down or impairing memory. So what you have is a drug here that impacts this gene. This gene is then transcribed. As it's transcribed and translated, it then makes this protein that blocks these channels. So this has a huge effect, because if you didn't have the drug, the COX-2 gene would not be transcribed or translated, and then you would not have the memory impairments as described here. Okay, so let's turn our attention here to cells and organelles. One of the major things you're going to need to know is the difference between a prokaryotic and eukaryotic cells. Prokaryotic are our bacteria cells. They have no organized cell organelles. They do not have a nucleus. They do have genetic material and DNA. They have cell membranes, and they have a structural different cell membrane and a cell wall. We have plants, which have a cell wall, a nucleus and organelles, including chloroplasts. But they do not have centrioles, and these plants are covered with cell membrane. The cell membrane in the animal cell is very similar to the plant cell. It has a nucleus, an endoplasmic reticulum, as you see in both plant and animal cells, and mitochondria. It also has centrioles that are going to be important for cellular reproduction. And they also have organized cell parts called organelles, and those are listed here. So the two major cell organelles I want to talk about are this concept of energy and the movement of energy between photosynthesis and respiration, or in a sense between chloroplasts and what else? Oh, chloroplast and mitochondria, sorry. Okay, so let's talk about energetic enzymes first. So here's the enzyme pathway, and what you see here is substrate concentration versus enzyme activity. The normal enzyme you see is here. This is the rate. If there's a competitive inhibitor and a non-competitive inhibitor, could you predict which of these two lines are which? I'll wait for a second. 1, 2. If you guess correctly, this is the competitive inhibitor, and this is the non-competitive inhibitor. And the reason you see it in this line is these two enzyme reactions, the substrate and the active site and the competitive inhibitor, are all competing for that same active site. So you're going to have these two lines much closer. If a non-competitive inhibitor blocks the active site, you're going to see a change much faster. In this case, you see the optimal range of enzymes is a quick review. In this case, this is temperature. And with the role of temperature, you could see that you have these different enzymes have different optimal ranges. We could change the temperature to pH, and you would see again different enzymes with different optimal ranges. We talked briefly about these enzymes in this thing called negative feedback, and I want to talk a little bit about two types. One, of course, is regulating the enzyme directly. It's on this side, or I can regulate the genes that are then transcribed and translated. And so in either case, I can either block some part of the enzyme causing a change, or I can actually affect the gene transcription and translation process that makes the enzyme. Quick review over here. Here's your normal enzyme substrate complex. The competitive inhibitor, the non-competitive or allosteric inhibition, and then this concept of feedback. Take a minute to go ahead and take a look at photosynthesis versus respiration. And this is aerobic respiration. You should be aware of the differences in how you can change any one of these reactants. That would be in photosynthesis, water, carbon dioxide, or light, and how that would affect the output. And in aerobic respiration, it's the carbohydrates and the presence of oxygen, as well as the limited number of nicotinamide adenine dinucleotides, that's the NAD, in the cytoplasm that can regulate respiration. And below are the two processes that show this. Okay, so this was a very quick overview. Just wanted to introduce you to these, and this is the highlights and the important parts. Stop it at any time if you feel you need to go back over and flip back. All right, thanks.